Alan Rockefeller is a student from Genbank, and he's done a quite extensive amount of traveling since the last time he's been here in February for this year. But we're really excited to hear about this topic because it's kind of a teaser for our foray that's happening later this kind of summer. So let's give a round of applause to Alan Rockefeller.
If you guys have any questions, just go ahead and shout them out. Feel free to interrupt me mid-sentence. I'll take questions at the end too, but it's really better if you ask questions right when you're thinking about them. So I've been hunting mushrooms in Baja for probably 5 or 6 years now, and there are really good mushrooms in Baja, California. And so here is where you find the mushrooms. All right, here's where you find the mushrooms. And so this is a map of all the agaricales from iNaturalist. And so there's a lot right around here. That is the Sausalito area. And then a whole bunch in San Pedro Martir. And then there are kind of like desert mushrooms scattered in other places too. And then in Baja California Sur, most of the mushrooms are Sierra La Laguna. And that is a really cool place. I'll talk a little bit more about that later. But zooming in a little bit, you can see Ensenada, the hills around there have a lot of coast live oaks. So that's all really good mushroom habitat. And then there's some mountains here. This is Parque Nacional Constitución 1857. And I've never been there, but it's mountain habitat. So it's got like white fir and all sorts of pines. And then San Pedro Martir down there is a really cool place. That is the furthest south that you find Abies concolor, which is the white fir, a really good tree for mushrooms. And also sugar pine, Pinus margretiana, grows there as well. And so here's what most of the mushroom habitat looks like in Baja. This is a little bit south of Ensenada here. And you can see it's all chaparral up in here. And then coast live oaks down in the canyons. And you'll find really good mushrooms in both habitats. Though, most of the big mushrooms will be with the coast live oaks. And that's what the coast live oak habitat looks like. And when I went down there this time, I went with Sergio. He was here in the audience today. Really cool guy who lives in Ensenada, and he's been finding a lot of mushrooms. And I met him on iNaturalist. I noticed he was posting a lot of mushrooms on iNaturalist and collecting them and adding the microscopic features as well. So that got my attention. And he's also saving all of them. So one day we'll get them all DNA barcoded. And then I also went with Joey, who makes all the crime pays but botany doesn't videos. And so if you don't follow him, he's a really cool person to follow. The videos are extremely entertaining because every other word is profanity. And he has this persona as kind of like a mobster, and all this mob boss wants to do is teach you about botany and mushrooms. So super entertaining person to travel with as well. And so I've been going to Baja for many years with Joey to look for rare plants. And recently he's gotten into mushrooms as well. And one cool thing about Baja is that you can camp out pretty much anywhere. It's not like the United States where someone's going to be like, get off my land or whatever. So we just kind of set up a tent wherever seems like a reasonable place to do it. And that's a couple of places that we camp. This place here is really cool. This is, I think it's called like Sendero Los Coyotes or something. It's a little bit south of Ensenada. But we've been going there for several years. I think I have a couple more slides about that in a bit. There's really cool plants in Baja as well. This here is a bujum tree. And so bujum trees are crazy looking trees. Looks like they're something out of Dr. Seuss. And you see these are on the El Rosario area. We also found some psilocybin mushrooms in this area as well. Those didn't make it into my talk, so I'll just tell you about them. But they are gymnopilus, and they were on some of the kind of desert chaparral stuff. And there's a little gymnopilus, kind of looks like Gymnopolis saponaeus. But we picked them, and after a while, we noticed that the stem bases were staining blue. And so that's the psilocin polymerizing. And so even in these kind of desert dry habitats, you still find all sorts of cool mushrooms, including psilocybin mushrooms. Another really cool plant that I like to see in Baja is called Insilia densifolia. And this plant is extremely rare. The native habitat for this plant is only about 100 feet long and grows nowhere else in the world. So this is the spot here. It's really hard to get to. We had to drive up this wash for quite a few miles to get there. And then once you get there, there's like a little shrine, shows the little location. And then another really cool one is Insilia ravenii. And Insilia ravenii only grows on one kind of little granite mountain in the whole world, and that's over in the San Felipe area. That's the spot right there. And so, you know, I really like to look for both plants and mushrooms when I go to Baja. And one thing that I found a lot of in 2019 was morels. And so this is Morchella rufibranea, which is the morel that you get with landscaping. And this cluster is just like a really dense cluster of them. And I found it right here. So there's an ensenada here, and then the morels were right there. And that is the place that's like Sendero Los Coyotes. And so zooming in a little bit more, Sendero El Coyote is the exact name of the spot. And zooming in even more, this is where the morels were. And you can see that they have turned it into a farmer's field now. So the morel spot is gone. But we went out there just to look for them in this February. And after a few hours, this guy came up in kind of official looking truck, and we're like, oh man, we're getting kicked out. And he's like, hey, and then he was actually really nice. He's like, oh, you should go up here. And so we followed him. And just a little bit up the hill from where we were, there's a beautiful kind of picnic area where you can have events and stuff. And then there's a trail, that's the Sendero that starts there and goes up the hillside, beautiful trail that kind of follows the canyon and stream. And there was a ton of really good mushrooms there. One thing we found there was Amanita villosa, which is the world's most delicious edible mushroom. And this place was the furthest south that it had ever been recorded. Another thing that we saw there is this Paneolus. And this Paneolus is a new species of Paneolus. It's closely related to Paneolus cinctillus. So almost certainly has psilocybin in it. And this is growing like right in the middle of the trail here. So this is Sergio photographing the Paneolus. And we found this actually in a lot of places. It was one of the more common mushrooms out there. I found it in like 6 or 7 different places. And I'm pretty sure all of them are up on iNaturalist with the exact coordinates. So if you're out there, you should definitely take note of the coordinates and go look for this Paneolus. There's a kind of the same thing, but like a bigger patch of it that we found. And yeah, this kind of grows right out of the kind of gravel grass area. Let's see. So what really needs to be done to figure out the mushrooms of Baja California is to do DNA barcoding on them. And so far, there's only 7 DNA barcoded mushrooms on iNaturalist. Whereas in California, we have 7,000 DNA barcoded mushrooms on iNaturalist. And these are all where they all come from. But you can see we have 1,726 species of fungi that have been DNA barcoded. So DNA barcoding is completely revolutionizing our understanding of California fungi. And it's so much cheaper and easier, or so much easier and faster than microscopy, and way more effective too. So I still use microscopes a lot, but I only do microscopy after I've finished doing the DNA sequencing. So these are the mushrooms that have been DNA sequenced from Baja. The Agaricus californicus is the same one that we get all around here. And then this desert thing, the Montagnia arenaria, this one is kind of like a desert shaggy mane. And then the Paniella cinctulus is not really cinctulus, but actually a new species, which also turns up in Arizona. Pretty sure that one doesn't have a name yet, but it's a sister species to Paniella cinctulus. And then you got this bolete, the Xerocoma subtomentosus. So that's the suede cap bolete, and that's the same one in Baja that we have all up and down the west coast. And then Cantumyces rosellis, we call it Cantumyces rosellis, but it's actually a new species because Cantumyces rosellis was described from France, and the DNA sequences that we have from California and from Baja do not match the European ones. Then we got Amanita, that's in the Novanuta group, but it's a new species. And Cortinarius, and this Cortinarius, I figured I'd never get it identified, but it's, I got pictures of that in a little bit, but Cortinarius albidolilacinus, really big phlegmaceum type thing that grows under coast live oak. One really cool thing is that Stephen Russell has offered free DNA sequencing for all California mushrooms. There's no limit on how many you can collect. And so really any mushroom you photograph, you should put the photographs on iNaturalist and then label it with the iNaturalist observation number and send it to Stephen Russell. And he will DNA barcode it using Nanopore for free. And then the mushroom will be saved forever in the University of Michigan herbarium. And so if you discover new species, which you will, and you have the holotype there, and you can name it. And then the DNA sequences go directly onto iNaturalist and also into Genbank. So it makes like a permanent record and really helps out with figuring out what mushrooms we have. So nanopore sequencing is this new kind of sequencing. And there's two very cool things about nanopore. One of them is that you can, the whole thing is run right on your desktop, so you don't have to send out the PCR product to another lab. You just get the results right there using a device that's about the size of a thumb drive. And then the other cool thing about it is that when you are doing a run, it reads like a couple million base pairs. And so it costs the same amount of money to sequence one mushroom or 960 mushrooms. And so if you're doing runs where you take 960 mushrooms, run PCR on all of those, pool the PCR products, feed them in the nanopore, then it only costs like 15 or 20 cents per DNA sequence. Whereas if I do it in my lab at home, it costs me like $6 per DNA sequence. And just a few years ago, it was extremely expensive. So the nanopore is not really practical unless you're running a huge amount of samples. So that's why we send all of our mushrooms to Stephen Russell, and then he just runs all of them. Oh, yep. Sorry, I can't really see when people raise their hands, so just start talking.
What's the preferred method of DNA extraction? For like, is there an all-encompassing one, like a phenol chloroform that I do, or?
Yeah, that's a really good question. What's the preferred method of DNA extraction? And it really depends on what you mean by preferred, but what we do is we use this product called IBI XAMP. And XAMP is really cool because you just take like 1 milligram of the mushroom, just a tiny bit of the gill, throw it in a tube, heat it up to 80 degrees Celsius for 10 minutes, and then the DNA extraction is done. So it's extremely easy, it's very cheap, and it's very fast. And that works, that gives us about 80% success rates. And then if you use a really nice DNA extraction, now you can use phenol chloroform, that works great, but the chloroform is kind of toxic and the phenol is kind of toxic, and then you got to deal with the toxic material disposal, and it's kind of hard to get. and it kind of takes all day to do those kind of extractions. So usually when we want to do a really good DNA extraction, we use something like there's a bunch of these different kits, but I think the one they use now, it's, I think there's DNEZ is one of them. I can send you the links to the really good kits, but those, what's that? Yeah, that's a good one.
What about the mobile?
Which one?
Mobile soil?
I bet any of those would work. I haven't tried that one. But there's one that we use for full genome sequencing that works really well for high molecular weight DNA, and that works really good for PCR2. And if you use a kit like that, you will get just about 100% success rate. But you know, it's a lot more expensive and it takes a lot more time. So we pretty much always just use this really quick XAMP thing, and then if it doesn't work, just rerun it, and that works pretty well. So this Cortinarius albidolimacinus, this one I found it in the hills just east of Ensenada, and all those beautiful coast live oaks. So really cool, really big chunky cort with the abruptly marginate stem base there. And I figured I would never be able to identify it, but it got 100% DNA barcode match to the holotype. I think this one was described from Southern California, but it goes all up and down the coast. And there's quite a few really cool Cortinarius that grow with these coast live oak. And here is the Contumyces rosellis. And this is one that I found a little east of Ensenada, and I DNA barcoded it in the laboratory in my garage. And turns out that it has the code name Contumyces rosellis CA01. So every mushroom, if we can't figure out exactly what it is, then we put a code name on it. And that way, at least we have a name to call it. And there's two of these, there's CA01 and CA02, and only CA01 has been found in Baja, but it's almost certain that CA02 grows in Baja as well, because it was found just a couple miles north of the border. But really cool mushroom if you look at it really close. And then this one is Amanita species CA03. And this one, usually people call it Amanita novanuta. However, the real Amanita novanuta, we know what it is because we have a holotype sequence. And so this one, the DNA barcode is quite a bit different. Turns out this one is really common around here. It's way more common than the real novanuta. And then as you start going further north, the real novanuta gets more common. And this one becomes more rare. But this one's pretty common in Baja and really common around Escondido area. And so it stains red where damaged. And so you can see it's like blood red, and it's a pretty good edible. Probably a little bit poisonous raw. But if you cook it, it's really good. And that's the iNaturalist observation. You can see I put the DNA barcode in all of the iNaturalist observations, which is really nice because then you can search iNaturalist observations only for the ones that have this DNA barcode ITS observation field. And so if you want to know what something really looks like, instead of just stuff that maybe is misidentified, you can just do a search for the DNA barcode ITS stuff, and you can see all of the sequence stuff come back. And then you can also see that we put in the provisional species name. And so you can search on the provisional species names. And you can do that just by clicking on the provisional species name, click observations with this field and value. And now these points on the map are the ones, is that CA03, that new species of Amanita. Another one is this Xerocomus subtomentosus. So this is the suede-packed bolete. And occasionally it stains blue, not usually, but it's the same thing all up and down the coast. And then here is the world's most boring middle ground mushroom. It's
Sorry, all the way up and down the coast, starting where to where? Like where you just showed us on the map, like just in like lower California down to part of the Baja, or do you mean the whole thing? The
So
whole
that one is a coast live oak associate. And so it's not going to go much further south. It's not going to go any further south than the coast live oak goes. So, once you drive about 4 hours south into Baja, the coast live oak stops. And that's probably where that mushroom stops as well. And then north, the coast live oak goes just about to the Oregon border, but then in Oregon you have Gary oak. And so maybe it grows with Gary oak as well. I would have to look it up and just look up the distribution map of that, and then we can see it exactly where it grows. So this one is so boring because it's in every person's yard. But Agrosome pediates is a European name, but it's the same mushroom all over the world. So it's the same mushroom in California and Baja and Russia and China and everywhere else. So there's hundreds of DNA sequences of this one on Genbank, and this sequence that I got from Baja, California matches all of them. This one is really cool. This one is one of the red tubarias. And we have a couple different red tubarias in California. So it's probably Tubaria punicea or Tubaria vinicolor. They look very similar. But they are bright red tubaria, has a rusty orange spore print. And this one we found on a stump of Baja bird bush, which is the Ornithostaphylus. And then got Ophalotus olivascens, and around here you find this one on coast live oak and also eucalyptus. But in Baja you find a different host. So this one was found on Meloisma laurina, which is the laurel sumac. We found it on a few different things. This one is cool because it glows in the dark. So if you take it into a dark room, it actually makes its own light. And it's poisonous, and it has a lot of dye in it. So it's really good for making watercolor paints and for dyeing fabric. So the common name of that is the jack-o'-lantern. You'll probably see it all the time. And then this one is one of the world's most boring mushrooms. Some of my friend Jacob Polk, when he was in the San Diego Club, it was so boring. We call it Stereum hirsutum, but it's so boring that he would just call it stir because it's on every single log. But the cool thing about it is that the scientists have not discovered it yet. So it has no scientific name, and Stereum hirsutum was described from Europe. But it's kind of cool that the most common mushroom in California still has not been named. And then here's some clitosebes, and clitosebes are super difficult to identify because there's a lot of them, and it's very difficult to figure out until the holotypes get sequenced. We're really not going to have any clarity on the clitosebes, but that is Sergio photographing these clitosebes under some coast live oaks. Another extremely boring mushroom is Vulvoputeus gloriocephalus. And this one is cool because it hatches out of an egg. So first it looks like a puffball, and then when it gets bigger, it looks sort of like a destroying angel, which is deadly poisonous, except that these have pink spores, so the gills turn pink when it starts to mature, and it's edible. This is the Amanita ocreata. So this one is the deadly poisonous lookalike. And this one was pretty common on the coast live oak. Also hatches out of an egg and has this kind of like sac-like vulva, stem base. One difference is that the gills never turn pink in this because this is a white spored mushroom. Then another difference is that these have a ring on the stem, whereas the Vulvoputeus never has a ring. And then here is the world's most delicious edible mushroom, Amanita villosa. So that's called the springtime Amanita. And this one we found on that Sendero El Coyote trail. And that's the furthest south that it's ever been found. It's mostly endemic to the state of California, but it's turned up a couple times in Oregon. And it gets pretty common in northern Baja. But really cool little grisette. It tastes a little bit like scallops. I think it's my favorite edible mushroom to eat. It looks a lot like Amanita ocreata, so you definitely have to know what you're doing before you start eating them. But they're actually pretty easy to tell apart from Amanita ocreata if you know what to pay attention to. One difference is they never have a ring on the stem. And here's Joey, makes the Crying Pays Botany Doesn't videos. He's photographing the Amanita ocreata off of the El Coyote Trail. You can see he's using a flash diffuser here, And so with his older photos, he would just use flash on everything, and there was this really harsh shadow. But by putting this white bib on his camera, then the flash hits that, and it makes the shadow a whole lot less harsh. So his photos look a whole lot better now that he starts putting this on there. Another one that's pretty cool is Amanita calyptrotoides. And this one is pretty common around here, but this one is Amanita section Caesariae. So section Caesariae also has like the Cocora and Amanita Caesaria. And this one is called the candlestick Entoloma because, or candlestick Amanita, because the stem kind of looks like it's made of wax. There's no other Amanitas that have that stem texture, but it's kind of a little Amanita with a striate margin and chestnut brown cap color. And so this one's also mycorrhizal with crosslight. And then this one is kind of cool. This is Amanita sponsis, and I put sponsis in quotes because it has not been formally published yet. But this is really close to Amanita novanuta, which stains red. However, this one stains brown. And then this particular one has a crazy tetraploid mutation. So it just has these bumps instead of gills. And that's pretty rare in this species. Usually they just have gills like a regular Amanita, and it'll just look like a novanuta that turns brown. Another thing that you see in only one place in Baja, California is Amanita muscaria. And this only grows in San Pedro Martir. And so San Pedro Martir, it's a 4-hour drive from San Diego, but such a cool place. We zoom in, here's where the Amanita muscaria is found in San Pedro Martir. That one grows with conifers. This one here is a little Galerina, and it's kind of little thing found on a hillside. And then there's a lot of very cool entelomas in Baja. And entelomas have a lot of blue colors very often. This one kind of has this steel blue stem. But you can tell it's not a psilocybin mushroom because no matter how much you damage it, it will not turn any bluer. But they got pink spores, and there's a whole bunch of them. So this is a really difficult group just because there's so many and they're really hard to tell apart. One of the coolest habitats that we found was under this Baja bird bush. So it's called Ornithostaphylos oppositifolia, and it's endemic to Baja, California. And this is a bush that is in the Ericaceae, which is the blueberry family. And something cool about the blueberry family is that it can have mycorrhizal mushrooms growing with it. So here we got a bunch of geopora, and that's the Baja California bird bush there. But it's just a little shrub that grows out in the chaparral. And so you would not expect to see amanitas and russula and lactarius and all sorts of things under it. But we found all of those things just under this shrub out in the chaparral. This one here is an undescribed lactarius. And you can tell it's lactarius because it has the zonate cap, so kind of concentric zones of color in the cap. There's not many mushrooms that do that. And then we found some russula. This is something around russula cremora color. And then this is lactifluus, and this is something that I have not noticed in San Diego. But lactifluus is extremely spicy. So you take a bite out of it and it just burns your tongue, just like you took a bite out of a habanero pepper. But it has kind of milk that comes out. So this is in the lactifluus piperatus group.
Is that the same compound that's in spicy peppers?
No, it's different. So spicy peppers have capsaicin. And this one, the russulas and lactarius have sesquiterpenes. So yeah, similar effect, but completely different molecular structure. And then the sesquiterpenes are not heat stable. So if you cook them, the toxin goes away and it makes them edible. So here is some geopora just coming out of the ground. And then here's that psychoactive paniolus again, just right on the side of the trail. So here's a close-up of the lactifluus. And usually it doesn't do this with the crazy gills going down the stem, it's just that one. And then Coltricia is the only ectomycorrhizal polypore. So these polypores can only grow with these trees, and these particular ones were growing with the Baja birdbush. Coltricia is one genus that has not been studied much, hardly at all in North America. So we borrow the European names, and I think mostly the European names don't really work, so we just misapply the European names until somebody names all of our Coltricias. This one's probably super rare because it probably just grows with that Ornithostaphylos shrub. And then the Lephora, these are cool because they have spores that look like little crumpled up paper balls that you might throw at your teacher. But they're also mycorrhizal. They're also growing with the birdbush. And this thing was a Tylopolis. This one, I think Sergio found this one, and really cool bolete. The Tylopolis are really rare. On the West Coast, we have just a few species. Pretty sure this one doesn't have a name yet. But half of the Tylopolis are super bitter, and then the other half are sweet, or just kind of like mild flavor. This one had a real slight sweet flavor, and so if they're not bitter, they're edible. And then we also have a bunch of desert mushrooms. So this one is Podaxis. And so this was out by the Encilia ravenii spot. And they're like puffballs that grow on sticks. And then if you cut them open, you can see the spores. So they fruit when it rains, and then they just start distributing the spores, whenever the wind blows, for a very long time. So they're uniquely adapted to the desert. And then this is the Montagnia. And so it's very closely related to a shaggy mane. And these, I sequenced the DNA, but I couldn't really figure out which Montagnia it is because there's not any good reference data for that. So I know that the same Montagnia grows all over California, in the Baja as well, but I don't know if it's the one that we call Montagnia arenaria. Geographically, I think it's in the right part of the world to be that species, but there's several Montagnias. Somebody needs to disentangle these. And then here are the Tulistomas, and this is a really common desert mushroom. And again, they're puffballs on sticks, but they have this cool ostiole which releases the spores up there in the very top. And you see these a lot of times just kind of out in the desert. And if you're looking for mushrooms in the desert, the best trees to look under are the mesquites. So the mesquites are kind of one of the few trees that drop a whole lot of leaves and create leaf litter, enough leaf litter to support the fruiting of mushrooms. We also see these under coastal live oak. So here is just kind of like zooming into the Ensenada area, and all of the pink dots are where people found guild mushrooms. And so Ensenada is a really nice place to hunt mushrooms because there's so much good coastal live oak habitat nearby. And so you can see this area is particularly good. I think this is where Sergio took us. There's a lot of good coastal live oaks there, and there's also a lot of Tecate cypress. And so Tecate cypress is also a really good tree for mushrooms as well. Here's a Helvella. It's like Helvella compressa. So pretty cool looking Helvella, should be edible. Coastal live oak. And then this one is pretty common around here too at the beginning of the mushroom season. And this is Swellellus amygdalinus. So it's a really big bolete with bright red pores, and then the pores kind of turn orange as it matures, kind of like a suede type top. If you look at the stem, it does not have the reticulation pattern on the stem. It's just kind of like dots. And I've heard that this one is poisonous if it's raw, but if you cook it, a bunch of people are starting to eat it. And so it's not even, it's very distantly related to the Satan's bolete, which also have red pores. So people used to think that all of boletes with red pores are poisonous, but actually there's a bunch of different genera of boletes that have red pores. And the ones that ended up in rubra boletus are the ones that are poisonous. And then this one, distantly related over in Swellellus, and is pretty good to eat. These are really cool because you cut them open and they stain a beautiful royal blue color. They stain extremely quickly, like within a second. So it's some of the best staining reactions of any of the mushrooms out there. And then this one is Areobolides flaviporus, and call it flaviporus just because of the super bright yellow pores. But sometimes the pores run down the stem a little bit and make it look really cool. But this is another one that you'll see under coast live oaks in Baja. And then this is one of the most common boletes, Xerocamelus dryophilus. So dryophilus means oak loving in Latin, or maybe it's Greek. And this one, you can always tell it because it has a two-tone stem. So the upper part of the stem is yellow to white, and then the lower part of the stem is red. And often gets that hypomyces on it. Hypomyces microspermis likes this one a lot. They don't stain blue very much. They stain blue a little bit, and they're a reasonable edible. No, so this blue staining is caused by variegatic acid, and variegatic acid is completely different molecularly than psilocybin, and you can tell that it is not a psilocybin mushroom because when you touch it, it stains blue, and then after about 10 minutes, the blue goes away. Whereas with psilocybin mushrooms, the blue staining is caused by the psilocin polymerizing, and it's a permanent blue reaction, so it stays blue for years. Another really good bolete that you find in Baja is Boletus borosii, and you'll find it on the coast under oaks or at high elevation under the conifers. But Boletus borosii is one that's in the Porcini group, so it has the kind of net-like reticulation in the upper stem. And these are like really big, massive, firm mushrooms, some of the best edibles. So far, nobody has found Boletus edulis bar grandigelis, that's the other coastal porcini in Baja. It's likely that it occurs in San Pedro Martir. And another one that would be really good to look for in San Pedro Martir is the spring king, Boletus rexvaris. And all the right trees are there, but I don't think anybody's really been out there in the spring. But I think the right time to go would be like late May for that. So if you want to do something good for the mycology, you can just go out there and hunt mushrooms in Baja and make iNaturalist observations with all of that. Because when we're making talks like this or just figuring out about the mushrooms in Baja, we use the iNaturalist observations to figure out what occurs there. We also get a lot of entelomas in Baja. This is the really big entoloma that's super common under coast live oak. And this one, you can call it Entoloma livido album or Entoloma ferruginans. There's actually a lot of different species. There's way more species that look like this than have names. So it's a big species complex that needs to be untangled. But sometimes they smell like bleach. Sometimes they smell like perfume, usually a little bit of both. And they're rumored to be poisonous. I'm not sure if they really are. But like all entelomas, they have pink spores. So the older mushrooms, they start to get pink gills. And then here is the Gymnopus dryophilus. And Gymnopus dryophilus, I sequenced this one and it matched European sequences. So it's a European name, so I think this is the real dryophilus. But it's an unexpectedly good edible mushroom. And dryophilus means oak loving in Greek, so this really likes the oak trees and is super common. And then here is Agaricus campestris. This one kind of likes grassy areas, and you can always tell because the stem base kind of like tapers to a little point there, and then it has a ring, but not much of a ring, and the ring falls off really easily, and it's a very thin ring. One thing you'll notice about all of the poisonous agaricus is that they smell bad and they all have very thick rings as if the ring is made of felt, whereas the edible agaricus have very thin rings and they do not smell bad. So this one doesn't really stain at all. And then we got Agaricus californicus. So this is one that's a member of the Lose Your Lunch bunch. So it'll make you throw up. There's a chemical in it called phenol, and phenol is toxic and it also smells bad. So you can smell the toxin in this mushroom. And the best way to smell it is just to scratch the stem base and then smell that. And then Agaricus californicus, if you scratch the cap, the cap does not stain yellow at all. But the stem base does stain yellow. So you cut it in half and then only the stem base will turn yellow. And this characteristic is called fleetingly lutescent. And what that means is that they stain yellow, and then after about 10 or 15 minutes, the yellow turns brown. If instead the agaricus stains yellow, but after 10 or 15 minutes, the yellow stays there, and it stays there forever, even when it's dried, just kind of like maybe fades to more of a gold color, then that is one of the edible groups.
Yeah.
Yep.
You know, the
It's like the actual molecule phenol.
Yeah.
So I'm not sure exactly what phenol red is, but maybe it has phenol in it. It's probably a mixture of like a red dye and phenol, but yeah, it's probably the same molecule. Yeah, it's actual phenol. So it kind of smells like a medicine cabinet. But not everyone can smell that, so you should learn to recognize it as well. This one, I was a little bit surprised because I sequenced the DNA and it came back as Agaricus californicus, but it was growing kind of more out in the desert and it was kind of clustered. But it's just a normal Agaricus californicus that was just kind of pissed off because it was in the desert. And then if you scratch the cap and it turns bright highlighter yellow, like instantly, then that one is Agaricus xanthodermis. And so that's another member of the Lose Your Lunch bunch, and see that in Baja as well. And so here you can see it's just like highlighter yellow, and after about 10 minutes, that turns brown. Another really cool thing that I've been seeing is the cystolepiota. And cystolepiotas are rare. This is the first observation of a cystolepiota from Baja, California. I'm a naturalist. But they're very fragile mushrooms. They got free gill attachment, and they have this really cool cap texture. So it's kind of like a powdery cap, like that. And this one had these cool water droplets on the top. But I wanted to show you a little bit about how I take these pictures. And so I'm gonna try to run over to the laptop and show you a little bit about that. See if we get crazy feedback. So I like to take the pictures using a technique called photo stacking. And so what I do is I take all of the pictures on my camera and I load them into Lightroom. And so here they are in Lightroom. And the photo stacking technique works where you take a picture and you can see the picture, this is focused right on the very front part there. And then I take another picture, it's focused a little bit further back. And so as I cycle through all the pictures, each picture is a very small slice of the final image. And so I find the image which has the very back of the mushroom in focus right around there. And so in this case, there's, I think about 80 pictures or something. And then I export those into some software called Helicon Focus. And what Helicon does is it combines all of these pictures into one final image. So here's that cystolepiota. And in this case, I have 23 images. And these are all taken automatically. Some of the higher end cameras that have a feature where it takes a picture and then changes the focus a little bit, and takes another picture, and just keeps repeating that really quickly. And if your camera does not do that, there's two ways you can do focus stacking anyway. One of them is to take a picture and then change the manual focus a little bit, and just take another picture and change the manual focus a little bit. And then the other way is to move the camera closer physically. So you can either use a focusing rail, or you can just like move the camera physically closer to the mushrooms. Sometimes I even like push my tripod forward. But the really cool thing about this technique is that you can bend the laws of physics and get as much depth of field as you want. And then I keep the aperture all the way open. And that way the background is really blurry. And so that way the subject stands out in the background really well. So if I just load these images in here and hit render, then you can watch it assemble all of these pictures into the final image. And there it is. And because the aperture is all the way open, you get really good detail. So you can like zoom way in here and yeah, get really nice detail. But then if you look, you see the background is very blurry very quickly. So I pretty much take all of my pictures using this focus stacking technique. And if I'm not going to use that, then I just use a cell phone. And the cell phones work really well for very large mushrooms. So the cell phones do not work very well for smaller mushrooms. So here's that paniolus. So this is that new species of psilocybin containing paniolus that we were finding around the El Coyote Sendero. And this one is 89 images. So it's taking a little bit longer, but you can start to see the mushroom coming into focus here. And now it's done. And you can see that the background is super blurry, so the mushroom really stands out to the background. And I like to start the stack pretty early, so the foreground is completely in focus. You get really good detail there. You get, you can see all sorts of details. So that's how I take all of these pictures. I'm mostly shooting JPEG. So if you shoot in JPEG, it throws away most of the data. Kind of depends on what camera I use, actually. So most of these are taken with a Nikon Z7. And the Nikon Z7 is such a good camera that the JPEGs are really good quality. And the JPEGs are so good that I do not even need to use the RAW format. I do use RAW. I don't get a whole lot of benefit from the RAW. But then I also have an Olympus OM1 camera. I got that camera because the macro lens for that camera is better. And that has a much smaller sensor. And so the image quality is not nearly as good on the Olympus. So I use RAW on the Olympus. But those RAWs fill up your hard drive really quickly. So
how long does it take to shoot 89 photos?
Well, that depends on how much light there is and which camera I'm using. On the Olympus, if there's a lot of light, it can do it in about a second, maybe 2 seconds, because it's really fast. I usually use this Nikon Z7, and that one does like, I think about 6 photos per second. But it also depends on the shutter speed. So I always use the aperture priority mode when I'm on a tripod, and I'll usually adjust the ISO. So the shutter speed is about 1/4 of a second. So 89 pictures, that's going to take 15, 20 seconds to shoot that. So if it's really dim, I'll turn the ISO up, so I'm not waiting like 5 minutes for all of the pictures to take. But usually I'll shoot at ISO 64 just to give it the best quality images that I possibly can. And yeah, it actually takes me as long as it takes me to find the mushrooms in the field when I'm at the computer after I've done mushroom hunting. So it's a lot of work to put all these things together, but the actual picture maybe takes 10 seconds to take, and then maybe takes me a minute or 2 to set up the mushrooms, set up my tripod, and then a couple minutes in Helicon to stack them. So if you can avoid all of this just by using a cell phone, that way everything is all instant. And what I'm using my cell phone, I take all of the pictures using the iNaturalist app, and that's really good for a couple of reasons. One reason is that it will save the GPS coordinates. And then the other reason is that I don't have to upload my pictures at the end of the day. They just, as soon as I get self-service, it uploads automatically. So usually what I'll do is take pictures of everything with my cell phone, and then the really good stuff or the really tiny stuff, I'll take pictures with the camera as well. And then because I'm usually mushroom hunting every day, I don't have time to assemble all of those pictures. But a lot of times, if I'm making a talk, I'll assemble the pictures, or once I get the DNA sequence back, then I'll go back and find the good pictures, put them online. Another really cool thing that I'll see under course live oak in Baja is Marasmius plicatulis. And these are really cool because they kind of have this iridescent sheen on the cap. You can sort of see it here, and that's caused by broom cells, or as I call it in Baja, California, células de escoba. But they actually look like little brooms, thousands of them. I think like the Disney movie, The Sorcerer's Apprentice. But it's like a real close up. You can see like they look really cool, kind of like interveno scales here. So I really like finding Grasmius platypus. And then this one was one of my favorite finds from last year. And this was 2 months ago in February. And this is from San Pedro Martir. And so we went there, we just kept driving until we saw coast live oaks. And they never had any lumber mills there. So there was no logging done in Baja, California. So most of the trees are old growth trees. And so there's absolutely massive coast live oaks. And that's really good because when they log, all those mycorrhizal mushrooms die. And it can take hundreds of years for them to come back. But there's all these big coast live oaks, and then there's also snow on the ground everywhere. So it's really interesting. We found these mushrooms just a few feet from the snow. But this was one of my favorite mushrooms. This is Portnarius from subgenus Leprosopy. And what was so cool about it is that under ultraviolet light, it glows extremely bright. You can see the projector is so bright, the projector is having trouble. But this whole subgenus of Portnarius glows extremely bright with ultraviolet. It's so bright that you can see it like by 100 feet away. And this is beautiful golden color.
Did you have the UV light to shine on it?
Yeah, so whenever I'm mushroom hunting, I always carry a couple of UV lights in my backpack. This is just a cell phone photo because I haven't even put together my good photos of this yet. But yeah, well, one cool thing about fluorescent mushrooms is that they don't have any evolutionary purpose. And the reason I say that is because there are no sources of ultraviolet light in nature where there's not way more white light to go along with it. So, the sun gives you lots of ultraviolet light, and it does make these look a little bit brighter just because it's converting the ultraviolet into visible light, but there's so much white light to go along with it that you barely notice it. But that's how fabric brighteners work, for example, is that convert the ultraviolet from the sun into white light, makes your clothes look a little bit brighter. But because there's no pure sources of ultraviolet light in nature, there wouldn't be any evolutionary purpose to being fluorescent. But what we're really doing with the ultraviolet light is we're using them like chemical sensors. So there are thousands of different chemicals in every mushroom. If you just run any mushroom through a mass spectrometer, you'll see hundreds and hundreds of peaks, and each peak is a different chemical. A lot of the chemicals have never really been researched or studied at all. So lots to learn here, but many of the chemicals in nature are fluorescent. So when you're shining an ultraviolet light on mushrooms, you're basically just looking for the presence of any of these different fluorescent molecules, of which there could be hundreds of different ones. And occasionally you can look up and see what the molecule is that's fluorescent, but usually we don't even know what the fluorescent molecule is. But it would definitely be cool to do some chemistry work and figure out which molecules are fluorescent in these things. So we're getting towards the end. I don't have a whole lot more for you because I have not done a whole lot of mushroom hunting in Baja California Sur. So this is the lower half of the Baja California Peninsula. And you can see, so this is a search for the agaricales, which are the gill mushrooms. And you can see there's a few gill mushrooms scattered around, but almost all of them are right at the southern tip. And the southern tip is one of the most special places in the world. This is called Sierra La Laguna. So up here is La Paz. If you want to go mushroom hunting here, you can fly into La Paz. And then, people usually fly in to go to all of these kind of beach resort areas here. But this mountain is absolutely spectacular because it's a beautiful, lush forest. And there are no other beautiful, lush forests for hundreds of miles in any direction, which means that there's tons of endemic species there. So there are hundreds of species of plants that grow in this forest here and in these areas around here and don't grow anywhere else in the entire world. And because of that, there's probably lots of mushrooms that grow only in this area and don't grow anywhere else in the entire world. Also, but very few people have really studied the mushrooms here. I've been up to this mountain 3 or 4 times, and all of the times that I went, it was really dry because I went during the mushroom season in California. So when I got all the way down to the southern end of this peninsula, it was the dry season. But the mushroom season here is the same as the mushroom season in southern Mexico. So that is like June through the end of September.
Sorry,
Yep.
let me ask. So you sound pretty familiar with it. Are you always driving and are there roads? Or are you stopping and then packing them?
So this particular mountain does not have any roads. And so you can drive up to about 600 meters elevation right to here, and then you leave your car here, and then you have to walk the rest of the way. It goes up to 1,800 meters. And when I went up to the 1,800 meter place, it's a place called Segunda Valle, Second Valley. And I went there because there's a madrone tree that only grows there and nowhere else in the world. So it's like Arbutus peninsularis, and it's really cool because that madrone tree kind of has whitish bark, unlike the reddish bark of the madrone right here. So they look almost like ghost madrones. And then there was also a pine that grows up there. It doesn't grow anywhere else in the world. Hundreds of other flowers and plants. But when I went, it was really dry, which was good because the trails were not muddy. If you go here during rainy season, the trail up there, it's really dusty when I went, but it turns to mud. So definitely have to watch out for that. And then I also was brought like camping and backpacking and sleeping gear. So I set up camp up there, and that's how I was able to have enough time to like hike all the way up this mountain, and then also look around when I was up there.
Sorry, so is it or is it just
Yeah, there's a pretty nice maintained trail that goes up there. There's a few hiking trails that are really popular. And I think they actually might close them during mushroom seasons, depending on how the rains are. So if the mushroom season is really good, you might have to sneak up there. But it would definitely be worth it because it's a very beautiful, very remote place. And one mushroom that I did find there was Hypomyces lactiflorum. So this is a picture that I took in Jalisco, right across the Sea of Cortez from Sierra La Laguna. But Hypomyces lactiflorum is a lobster mushroom, really good edible. But I just found this like little patch of bright yellow chip when I brought it back, and I DNA sequenced it, and it had the exact same DNA sequence as all of the other lobster mushrooms all over the world. So it was the real Hypomyces lactiflorum. So I know that grows up there. But if you go up there during mushroom season, sometimes I'll kind of just keep an eye on the iNaturalist for that area, and people are finding really cool things. A lot of the things I do not recognize from anywhere else, so it definitely needs more study and more people going there during rainy season and looking for mushrooms. Thank you very much. Any security concerns while I'm there by myself? That's a good question. And no, not really. Baja California is extremely safe. The rest of Mexico can get a little bit sketchy, mostly near the border. But the southern part of Mexico, where a lot of mushrooms are, is pretty safe. Definitely safer than Oakland or something like that. But yeah, there's really nothing that's going to get you out there. You definitely have to keep an eye out for rattlesnakes and stuff. I occasionally you'll see drug traffickers in Baja, California, but they're pretty nice and they'll just like, if you get a flat tire, the drug traffickers will come by and help you fix your flat or whatever. But there's not really a lot of crime once you get past Tijuana. So Tijuana is pretty sketchy. And you're right on the border. There's always a lot of crime, war on drugs. But once you get past that, then you know, I feel very safe in Baja, California. I feel real safe just setting up a tent wherever I am, camping out, and yeah, not too much is going to get you out there. You know, there's not a lot of animals will get you. Not too many people will get you either. So it's really relaxing. You know, pretty safe place. Yep. Question is it mostly Quercus agrifolia or the other Quercuses? And I find the Quercus agrifolia is a really good mushroom tree because it has the really big leaves, and so it makes lots of leaf litter. And also, there's just a lot of mycorrhizal mushrooms that grow with Quercus agrifolia. I have done a lot of hiking under scrub oaks, and I found a few mycorrhizal mushrooms under scrub oaks, but only about like 1% as many as I would find under Quercus agrifolia, the coast live oak. And so, pretty sure that Quercus agrifolia is the best tree for mushrooms, especially in northern Baja. But yeah, definitely look under the other ones, but you'll find there'll be a whole lot fewer and more far between under these other trees, other oaks. Yep.
Is the ITS sequence the main kind of barcode that you look for, or is there some other way you can?
So people have standard on the ITS sequence as the fungal barcode. And so ITS stands for internal transcribed spacer. And it's really good gene to use because there's like 100 copies of it in the genome. So it's really easy to amplify with PCR. And it's very variable between species. So you get really good species level resolution. What it does not work very well for is like figuring out how all the genera fit together because it's so variable that you won't get like good bootstrap support. You try to make a phylogenetic tree that includes a lot of species that are not closely related. So if you really want to figure that kind of stuff out, we usually do like a 5-gene data set. And that's with ITS, LSU, RPB1, RPB2, TEF1, beta-tubulin. And that gives really good resolution both at species level and also the genus family level. And we're also starting to do a whole bunch of full genome sequencing. That's pretty expensive. It costs us about $2,000 to do a full genome sequence on each mushroom. But I'm a member of a project called the Entheome Genome Project, and we're doing full genome sequences on all of the psychoactive mushrooms and plants and organisms in the world. And so we have about 20 of them sequenced so far. And we see really cool things when we do full genome sequences, like we can start to see that the psilocybin gene cluster is different in different psilocybin mushrooms. And that probably accounts for like the different tryptamines they make and the different effects and things like that. And you can make phylogenies that have really good resolution with full genome sequencing, but it is not very much to compare with yet in these full genome sequences. So ITS is nearly always the best gene to do. It's certainly always the best gene to start with. And then occasionally there's some mushrooms where they'll have the same ITS sequence and then sequencing another gene like TEF1 can tell them apart. Some of the Strobilurus and Gyromitra are like that. But 99% of the time, ITS sequence will answer my question. Also, you know, a sequence by itself doesn't tell you anything. It doesn't tell you anything until you compare it with all the other sequences. And there's so much reference data available with ITS that whenever I sequence some other gene, you know, I have a box of primers in my garage lab, and I got like 100 different primers in there, and I sort of sequence a bunch of different genes, but there's so little reference data in these other genes that I'm always really disappointed when I learned by sequencing these other genes, because there's just not much data to compare with.
Do you do the full ITS or just region 1 or 2 or?
I usually start with the full ITS, so I use like ITS1F with ITS4. And if that doesn't work, then I would use like ITS1 with ITS2 and ITS3 with ITS4 and separate PCR reactions. So that's just those separate genes.
So the reason I asked, sorry, is I've looked at the mycobiology and like so microbial communities like that living in
issue of form. But one of the things was it was very like it identifying things if we use one region versus the other, it like would find species or wouldn't find them, identify them. So I don't know what the
Yeah, and that's usually with the next generation sequencing. The
Yeah.
read length is so short that they have to pick one or the other. And I've had actually pretty good luck with identifying stuff with either one or the other. Like maybe 99% with full length, 95% it's just one or the other. So it works really pretty well. But yeah, it's definitely best to do both. Yep.
You mentioned that you were doing are you guys finding it wild? Are you guys cultivators
Yeah, the question is, are we doing wild psychoactive species for full genome sequencing, or is it cultivated ones? And we're doing both. We do a bunch of ones that we found in the wild. So we recently published a paper on psilocybe out in the forum, which is the most potent wild psilocybin mushroom known. And we did a full genome sequence on that and potency testing and just all sorts of stuff. That got published pretty recently. So that's a pretty cool paper if you're into that kind of stuff. But we've decoded the psilocybin gene cluster in that one. And we're doing a whole bunch of ones that I found in the wild, but we're also doing a whole bunch of cubensis cultivars. And it's kind of expensive to do it because we do both nanopore and Illumina. So we use the nanopore for the long reads, and then the nanopore just doesn't have the accuracy. So then we use Illumina for the accuracy, and then use those long reads from nanopore, the scaffold, to assemble the Illumina data. And it gives us a really high quality genome, but it's pretty expensive. So, about $2,000 each, we mostly get people that want to donate money to get things sequenced. And so if people are willing to pay for the sequencing, then we will sequence their cultivars. So we're able to start building phylogenetic trees of like all of the different cubensis varieties, and we can see like how they all were bred and that sort of thing. But for the most part, it's the wild ones that interest me the most. And most of the ones that we've done so far are the wild ones, but I think we're doing about 10 different cubensis strains this year as well. Yep.
Are you concerned about there being a loss of diversity on certain qualities?
The question is, am I concerned about a loss of diversity when cultivators cultivate psilocybin species? And you would think that it's a really big problem because people have been cultivating these strains that have just been inbred for like 30 years, but it actually seems like they work okay, even after like people are just growing out the same spores for many, many years. But what you can do is find some cubensis in the wild, or take a wild strain of cubensis and germinate one of those spores on agar, and then take your strain and germinate a spore on agar, so you have the monokaryotic mycelium, and then mate them, and then you'll get all of this genetic diversity from the wild back into your strain. And some of my friends have done that a little bit, and they don't notice a whole lot of difference. So it seems like the loss of diversity isn't really affecting things too badly. But I think also that cubensis is kind of a boring mushroom, and that it's not super potent, and it kind of hurts my stomach when I eat a lot of it. Whereas some of these other species, they're way stronger, and no matter how much I eat, I don't get a stomachache from them. So I think in the future there'll be a lot more people focusing on some of these exotic species like Zephyrine, and I think that'll be a good thing when those become available. Yep.
Are you also looking into gamma-induced stereo like muscimol and inorganic acid?
I haven't looked into the muscimol very much, but that's something that I would definitely like to. I think Jason Slott has sequenced some of those amanitas, done full genome sequencing. Jason Slott, if you haven't heard of him, he's a super cool guy. He's a researcher at the Ohio State University, and he is one of the people that discovered the psilocybin gene cluster, and he's also working with us on the Entheum Genome Project. And so pretty sure he has a full genome sequence of that. I could ask him when he's found out about the genes that make boostable ibotenic acid. But also Amanita lavandula in the Amanita citrina group. That one makes a tryptamine called bufotenine, and he has a full genome sequence of that Amanita, and he found the genes that make that tryptamine in that Amanita. So there's definitely a lot of cool genes out there, just scratching the surface. All right. Oh, yeah. You know, I've never seen Amanita muscaria in Baja. So what I did is I just went on iNaturalist and I found I made a list all of the mushrooms that have been found in Baja, and then I saw that it was there, so I just put one of my Amanita photos from Colorado in my talk. But those are found only at high elevation up in San Pedro Martir. So I think it's about, what is it, 3,000 meters up there?
Yeah.
And it looked like all of the muscaria points on the map. You know, there was a lot of them, and there were all of those pretty high elevation areas. So I would guess you find it like 2,500 meters to 3,000 meters. And also it'd be interesting to see like what time of year they're growing, because I was just in San Pedro Martir and I didn't see any Amanita muscaria. So maybe they're a fall mushroom, something like that.
I think you see them in July and August.
July and August. Okay, so they're with the monsoons.
Yeah.
So, you know, the same rains that hit Arizona and Jalisco and Oaxaca will also hit San Pedro Martir. And yeah, what you should definitely do is keep an eye on the weather. And when you see that San Pedro Martir is getting a lot of rain, then plan a trip down there. Because it's definitely worth going down there, but you don't want to drive all that way and just have it be dry. Yep. Yeah, the question is what algorithm and parameters do I use to align sequences? And I like muscle, and I just use the default parameters. But that other algorithm like Clustal W also seems to work pretty well. But I think more important than the actual algorithm and which parameters is that you manually kind of verify the alignment. And so after I've done this automatic alignment thing, I just go through and make sure that it looks like it did it right. You know, a lot of times, like one of the sequences here in alignment will have just one error in there, and it won't be like a real difference. You can just like delete that row out of your alignment if you're pretty sure it's a sequencer error, and you know, you can make the alignments pretty good that way. But yeah, just kind of like a manual sanity check is the most important thing to do with the alignments. Are there any good books about this? The only real book that I know about for this stuff is called Tree Thinking. And it's all about building phylogenetic trees. And it is very dense reading. So it's not an easy book to read. But it is really good. So so I think that's the one I would recommend. But most of the stuff I just learned from other people or just figured it out on my own. But if you want to learn about building phylogenetic trees or whatever, you just message me and I can like do a Zoom call or something, share my screen and show you how I built them. Well, thank you very much.
Let's give them a nice round of applause. Thanks, Alan. All right, so that was a lot of science. We're gonna throw another nugget of science in there with our science director, Marianne Hopp.
Hi, everybody. So that was pretty exciting. And raise your hand how many of you want to get involved? Yeah. And raise your hand how many of you have iNaturalist on your phone and use it? Woohoo! Okay, so that means that you can get involved in our citizen science project. If you go into iNaturalist and you look at projects, look for SDMYCO, SDMyco 2024. And if you join that project, then when you're out walking around and doing your mushroom hunting, you can take pictures and upload them to our project and help us build a map of the distribution of mushrooms in San Diego County. It's boundary restricted to San Diego County, so not when you're outside that area. But I just want to thank everybody who has participated so far, because we have 134 identifiers 31 observers.
Wow.
And we found 244 different species, and we have pretty much 1,500 observations. So, yeah. Please get out your iNaturalist, get outside and join in. Thank you.
So we're heading into our dry summer season, but we're looking forward to getting back into our rainy fall winter season coming up later this year. That's all I had, but we're gonna do a quick dance. If everyone wants to stand up. Clap your hands if you love mushrooms. Clap your hands if you love learning about mushrooms. And we're gonna continue clapping like it's the rain. Imagine the rain is hitting the ground. Make some sweet dance moves. And it's raining. I'll see you guys next time.